Hi, this is Dr. Johnson, and welcome to Chapter 6 of Vander's 11th edition of Physiology. I'll be instructing you in Chapters 6, 7, and 8 in this course, as well as Chapter 14 on renal physiology and Chapter 17 on reproductive physiology. Now, Chapter 6 is a fairly large chapter and will be a pretty complicated one due to all the neuroscience. However, having a good basic understanding of the nervous system is important in understanding how the body works, as you can imagine. Basically because the nervous system is involved in one way or the other with basically every physiological system in the body. Now, this chapter is broken up into 4 sections. Section A describes the basic structure and function of neurons, the various classes of neurons, other tissues that are found in both the peripheral and central nervous system, and how nerve cells grow when we develop in the uterus, and issues of neuron regeneration. Section B discusses membrane potentials, which is the basis of why electrically excitable cells like neurons, heart cells, muscle cells are able to generate action potentials. As you'll see, the basis for membrane potential evolves from the ability of these cells to separate negative and positive ions across their cell membranes, and then allow these ions to move across the cell membrane rapidly through specialized channels when the cells actually want to fire. Section C will look at the function of synapses, which are small but very complex structures, and as you'll see, synapses are in fact more complex than simply a space between 2 neurons, or a space between a neuron and a cardiac muscle or a striated muscle cell. Synapses are very interesting, and you'll find that section, I think, pretty. The information there will be very useful to you later in understanding how drugs work. Finally, Section D of Chapter 6 looks at the central and peripheral nervous system more as a whole entity, instead of looking at the separate parts and how they work, as was basically reviewed in Sections A through C. So the way I'd like to work in this course is to have you read through each chapter you're doing with me one time before you begin listening and looking at the PowerPoints for that chapter. So if you're able, please take the time to read through Chapter 6 at least once before you begin the PowerPoint. The PowerPoint that I make up for my chapters typically won't cover all the material in every chapter, but it's going to cover most of it, and will include the material that I think is most important to know based on my own experience with physiology. And also, the work required of you in this chapter also involves completing labs 8, 9, 10, and 14 on the Phil's Lab CD that you purchased along with your textbook. The labs are essentially self-explanatory, but as you hopefully read in the syllabus, you need to do the labs in the chapters that require them. You only have a total of 10 labs in this course, and not all chapters require you to do a lab. In fact, I only have 4 labs that I require throughout this whole course, and all 4 of them just happen to be in this chapter, Chapter 6. So, although you're burdened with 4 labs in this chapter, you won't have to do any labs for my other chapters, Chapters 7, 8, 14, and 17, when you get to those chapters. So, if you're ready, go ahead and move to the first slide in this PowerPoint if you're ready to start studying Chapter 6. Remember, don't try to do too much at once and get burned out. Work through the PowerPoints until you feel like you've had enough, and stop, take a break, and whenever you're ready again, just pick up where you left off. Good luck, and you have all my contact information if you need to reach me to ask me specific questions. Okay, let's start with a general overview of neuronal signaling and the structure of the nervous system. Communication by neurons is based on changes in the membrane's permeability to ions. 2 types of membrane potentials are of major functional significance: graded potentials and, more importantly, action potentials. Now, graded potentials are basically membrane potentials in neurons that are confined to a small part of the neuron, and typically are slow-acting potentials and don't spread along the axon of the neuron. Action potentials, on the other hand, are membrane potentials as you will see, in neurons that cause large changes in membrane polarity, typically spread along the axon of the neuron and occur very rapidly. You can think of neurons using graded potentials when they need to communicate over short distances, and action potentials when they need to communicate over long distances. A typical neuron has a dendritic region, an axonal region, and the dendritic region is specialized to receive information, whereas the axonal region is specialized to deliver information. As you'll see in the next slide, most neurons have what can be considered a receiving end for incoming information, i.e. the dendritic region, and an axon for delivering information to other neurons, usually by causing the neuron to release a neurotransmitter of some type into a synapse at the axon's terminal end. The two major divisions in the nervous system are the central nervous system, which is basically the brain and the spinal cord, and the peripheral nervous system. All the nerves that leave the central nervous system and innervate tissues outside the central nervous system, i.e. the brain and the spinal cord, are considered to be part of the peripheral nervous system. Within the peripheral nervous system, major divisions are the somatic nervous system and the autonomic nervous system, which has two branches, the parasympathetic and the sympathetic branches. So the somatic nervous system, as you'll see, is that part of the peripheral nervous system that controls voluntary movements in the body, such as those performed by skeletal muscles. The autonomic nervous system is that part of the peripheral nervous system that controls those activities that are generally carried out in the body without conscious control. For example, the autonomic nervous system affects heart rate, digestion, pupil diameter, among other functions. And most aspects of homeostasis are actually controlled by the autonomic nervous system. All right, when you're ready, let's go to the next slide. Okay, this is figure 6.1 in Vander's 11th edition, and it shows some of the basic structures of a neuron. It's important to remember when you're looking at this figure that this neuron is a single cell. This can be confusing to people taking physiology for the first time, so you sometimes have a tendency to think that all cells in animals are only round in shape. The figure on the left is a simple drawing of a neuron for reference, while the figure on the right in 6.1 is more what a neuron looks like under a microscope. In both cases, you should commit to memory the basic structures you see: the dendrites, the cell body, the initial segment or axonal hillock, the axon collaterals, and the axon terminals. In the classic form of communication between neurons packed together in the central nervous system, a single neuron may have hundreds or even thousands of axon terminals from surrounding neurons, forming synapses with its dendrites. These axon terminals from the surrounding neurons release chemicals called neurotransmitters into these synapses, synaptic spaces, which are in fact simply a synaptic cleft is simply a tiny space between the axon terminal and the dendrites. These neurotransmitters that are released into the synaptic cleft in turn will typically bind to receptors on the dendrites after being released into the synapse, and this binding provokes some type of response from the neuron whose dendrite receptors are being bound by these neurotransmitters. We'll be talking a lot more about this shortly in this chapter. As you'll also see, the response seen is typically one of two main options when a neuron is receiving neurotransmitters from another neuron. Option one is for the neuron to become what is called depolarized or electrically excited and generate graded or action potentials. Action potentials can be thought of as electric charges being generated in the neuron. As we'll discuss in subsequent slides, this electrical charge is created by changing the membrane potential of the neuron, and the charge typically starts around the axonal hillock and moves down the axon itself, terminating in the axon terminals and causing neurotransmitters to be released into the synaptic cleft from vesicles that are stored in that neuron's axon terminals. Again, we'll see a lot more of this in section C when we get to synapses. Option 2 for a neuron, if it's responding to neurotransmitters from another neuron, is for the neuron to actually become electrically quiet or hyperpolarized and not generate any kind of action or graded potentials. This should make sense since sometimes we need to have neurons be quiet and not communicate information to other parts of the brain via generating action potentials, while other times we do need them to talk with each other. So as you'll see later, neurons decide whether or not to generate an action potential by summing up the information they're receiving from hundreds or even thousands of other neurons that are forming synapses with them and releasing neurotransmitters. Some of these communications from other neurons will tell it to fire, while other neurons communicating with a single neuron are telling it to remain quiet. And this is the concept of excitatory postsynaptic potentials, or EPSPs, versus inhibitory postsynaptic potentials, or IPSPs, that we'll discuss in section C of this chapter. Alright, when you're ready, let's go to the next slide. Okay, figure 6.2 in Vander's 11th edition shows how neurons are insulated by myelin sheaths, which allow neurons to send an electrical signal down the length of their axon with much greater speed. Myelin's made up mostly of lipids, but also some specialized proteins. The main consequence of a myelin layer or sheath is an increase in the speed at which impulses propagate along the myelinated fiber, as I alluded to a second ago. You can almost think of myelin like the insulating material surrounding an electric wire. The myelin sheath prevents the current that's generated by the action potentials in a neuron from flowing across the membrane. And you'll see this complex process later. It'll make more sense to you. This forces the current to travel in a straight line down the nerve fiber to the unmyelinated nodes of Ranvier. These unmyelinated spaces at the nodes of Ranvier have a high concentration of ion channels, which, as you'll see, are important in generating action potentials. The incoming action potential stimulates these ion channels to propagate the action potential to the next node. Thus, the action potential jumps along the axon of the nerve fiber as it's regenerated at each node in a process called saltatory conduction. Along unmyelinated fibers, action potentials move continuously as waves, which is much slower than saltatory conduction. The importance of myelinated neurons can be seen in the so-called demyelinating diseases, which result in myelin being removed from neurons in both the central nervous system and the peripheral nervous system. Probably the best known of these is multiple sclerosis. A large part of the pathology of multiple sclerosis, or MS, is that the myelin sheaths of the neurons are broken down, probably by attack from the body's own immune system. If nerve fibers that carry sensory information become demyelinated, problems with sensation result. If nerve fibers that carry signals to muscles become demyelinated, problems with movement result, etc., etc. These are all symptoms that can appear in someone who does in fact have MS. Different types of cells produce the myelin sheath in the central and peripheral nervous systems. Oligodendrocytes make the myelin sheaths for neurons in the central nervous system, while Schwann cells perform this important task in neurons in the peripheral nervous system. Alright, when you're ready, let's go to the next slide. Alright, figure 6.4 in Vander's 11th edition. This describes how neurons are essentially divided into three major classes: afferent, efferent, and interneurons. It's important to understand the basic differences in these types of neurons. The central nervous system allows us to sense information about both our surrounding environment as well as the internal environment of our bodies that's outside of the central nervous system. An example of sensing our external environment comes, of course, from our five basic senses: sight, sound, touch, taste, and smell. This ability to sense our external environment is the basis of Chapter 7 in this book, which we'll discuss next. Of course, our bodies can also detect and then send information to the CNS about our internal environment. A stomach ache, a headache, pain from a kidney stone, gallstone, etc. are all examples of some painful pathology that's occurring inside our bodies, and information about this problem is being transmitted to the central nervous system. A lot of times, of course, we don't want it to be transmitted because it's very painful. All of the information transmitted into the central nervous system, either about internal body problems or external environmental issues, comes to the central nervous system via afferent nerve fibers. Afferent neurons have sensory receptors in our viscera, our muscles, our skin, our eyes, our ears, our tongues, etc., that sense the information needed about these areas and send that information to the spinal cord, which shunts it up to the central nerve, to the brain, for processing. Once the information is received by the brain, it's shunted from one area of the brain to another via the interneurons, which are neurons located within the central nervous system. They're also in the spinal cord as well as the brain, whose job it is to process incoming information from afferent neurons. If some type of response is needed to that incoming information from the afferent neurons, the interneurons will initiate a response in the appropriate efferent nerve fibers, whose cell bodies are located within the central nervous system. Remember, their cell bodies of the efferent neurons are located within the central nervous system. These efferent neurons will then initiate some response that will be sent out to the periphery to deal with the problem. A classic example of this is touching a hot surface with your finger. In this example, sensory receptors in your skin are quickly activated by the sensation of heat, causing afferent neurons to fire. These afferents send a message to your brain through your spinal cord that your finger is touching something hot. Interneurons interpret this incoming information and activate the appropriate efferent neurons in the central nervous system to deal with it. In this particular case, the efferent neurons that are activated would be motor neurons, which will release neurotransmitters where they synapse with muscle fibers in your arm, causing those muscles to contract and yanking your finger away from the hot surface. Incredibly, of course, all this will occur in just milliseconds. If you look at these afferent nerve fibers in figure 6.4, you can see that afferent neurons are anatomically different from the efferent nerve fibers. Afferent axons branch after leaving the cell body. One branch, called the peripheral process, is connected to the appropriate sensory receptor in the periphery. The other branch, called the central process, travels to the central nervous system, where it will bring information to the interneurons from these peripheral sensory receptors. It's important to note here that afferent neurons have no dendrites, and the cell bodies of the afferent neurons all lie outside the central nervous system. Conversely, almost all of the cell bodies of efferent neurons are located within the central nervous system and do have dendrites. An exception is the enteric nervous system, in which efferent neurons have cell bodies that are outside the central nervous system. You'll read more about the enteric nervous system in Chapter 15. So now please go to the next slide, which is Table 6.1. Take a few minutes to read the summaries listed there about these three types of neurons we just described. I put this slide in here just to go over some important terminology. It's important that you understand that when we talk about a nerve fiber in physiology, we're speaking about a single axon of a neuron. When we use the term nerve per se, we're talking about a bundled up group of neuronal axons in the peripheral nervous system. They can be afferent nerves carrying thousands of bundled afferent nerve fibers to the central nervous system, or they can be efferent nerves carrying thousands of bundled efferent nerve fibers from the central nervous system to the periphery. A mixed nerve is a bundle of nerve fibers that carries both efferent and afferent nerve fibers in it. Nerves are only found in the periphery, what we call nerves. Examples would be the vagus nerve, the sciatic nerve, the ulnar nerve, etc. So you need to make this distinction between nerves and nerve fibers. In the central nervous system also, bundled packages of neuronal axons are called tracts, not nerves. So within the nervous system we do have bundled packages of neuronal axons, but we don't call them nerves, we call them tracts. We only call them nerves when they're outside the central nervous system in the periphery. You can read about this a little bit more in your book as well. Okay, figure 6.5 is an introduction to the concept of synapses and how we define them. So I alluded to this earlier, and we'll talk about it much more in section C of this chapter, but synapses are very complicated entities, as you're going to see. The purpose of this figure, however, is just to teach some basic terminology that's used when describing a neuron and its relationship to a particular synapse. So when neuroscientists are describing the role of neurons in terms of how they interact with a specific synapse under a powerful microscope, for example, they typically use two terms, presynaptic and postsynaptic neuron. So look at this figure closely. I've labeled two of the synapses in this figure. One as synapse A and the other as synapse B. If you looked at synapse A and were asked which neuron was the presynaptic neuron relative to this synaptic or this specific synapse, you would hopefully say that the brown neuron is the presynaptic neuron in this case, which it is. The axon terminal from the brown neuron would be the one releasing neurotransmitter into the synaptic cleft here, and thus it's the presynaptic neuron when you're referring to this specific synapse. On the other hand, the green axon would be considered the postsynaptic neuron relative to synapse A, as its dendrites are the ones receiving neurotransmitter released into the synaptic cleft by the presynaptic brown neuron. On the other hand, if we were referencing the synapse labeled B in this figure, the green terminal would be the presynaptic neuron in this case, because it's the one releasing neurotransmitter into the synaptic cleft, and the purple neuron would be the postsynaptic neuron relative to synapse B. So the terms presynaptic and postsynaptic are used commonly, and they simply describe the relationship of neurons to a specific synapse, with presynaptic neurons releasing neurotransmitters and postsynaptic neurons responding to the neurotransmitters in some way. All right, let's go to the next slide. All right, figure 6.6 shows some of the roles of glial cells in the central nervous system. Glial cells act as support cells for neurons and account for about 90% of the cells found in the central nervous system. So that's interesting. You know, most of us, if you've never had a physiology course, have heard of neurons, and we tend to think that neurons actually make up all the cells in the brain and the spinal cord, but in fact, most of the cells found in the central nervous system are glial cells, and they perform many functions, especially in the brain. The different types of glial cells that you'll read about commonly, and you will read about in this chapter, are astrocytes, microglial cells, oligodendrocytes, ependymal cells, and Schwann cells. Some glial cells release glucose into the interstitial fluid around neurons for them to take up and utilize. Glial cells also vacuum up various waste products that neurons release into their environment. Astrocytes, a type of glial cell, are important in forming the blood-brain barrier, which prevents many potentially toxic or infectious agents from entering into the brain. The endothelial cells of arterial blood vessels entering the brain have very tight junctions between them, unlike the case of blood vessels in the periphery. This greatly restricts the ability of water-soluble substances to leave blood vessels and enter into the interstitial spaces in the brain. So in addition to keeping out potentially toxic compounds, this remarkable so-called blood-brain barrier also keeps large changes in blood levels of electrolytes, amino acids, or even glucose from being transmitted to the brain and causing electrical pandemonium. The so-called N-feet of the astrocytes helps to keep the endothelial junctions of blood vessels tight in the brain. This blood-brain barrier is also interesting in terms of drug development. If a pharmaceutical company wants to make a drug that works in the brain in some way, this drug has to be able to cross the blood-brain barrier and enter into the brain. So typically the way that's done is the company has to make the drug lipid soluble, because substances that are lipid soluble will diffuse across the blood vessels in the brain, diffuse right across them and enter into the brain. So when you see people, or you know of people that are taking drugs that have some type of effect in the central nervous system, the fact of the matter is a lot of time and money went into making that drug lipid soluble so that it could actually get to the place where it has to go to do its job. Microglial cells in the central nervous system act as immune agents, and they attack foreign compounds that do manage to get into the central nervous system. That's an important job since antibodies that protect the rest of our bodies from foreign material are typically too large to cross the blood-brain barrier and get into the central nervous system and work there. So we need something like microglial cells to do that job in the brain. These cells, these microglial cells, are much smaller than the other glial cells, and many of the microglia that are seen in the brain are resident microglia that migrated into the central nervous system during embryological development. Other microglial cells are derived from circulating blood monocytes, which migrated into the central nervous system a little bit later in life. Once they do migrate into the central nervous system, the monocytes take on the morphology of the resident microglial cell. Microglial cells are activated in infectious diseases as well, and neurodegenerative diseases, and other types of CNS injury will also activate microglial cells. All right, let's go on to the next slide. All right, let's go over this issue of neuronal growth. During embryonic development, stem cells evolve into various types of neurons and glial cells. These cells develop synapses with countless other cells in the central nervous system. Neurons and glial cells migrate through the CNS during embryonic development to some final position where they contact other neurons or glial cells and form synapses with them. This proliferation of neurons and glial cells is so extensive during our embryonic development that about half of these cells are actually pruned out by committing cell suicide, which we call apoptosis, to get the final number of CNS cells that we have around the time of birth or shortly thereafter. Neuroscientists aren't sure as to why our brains and spinal cord develop so many extra cells while we're still in the uterus, and then prune out so many of them by genetically pre-programming them to die after they've developed to a certain stage. But that's what apoptosis is, genetically pre-programmed cell death, whereby neurons or glial cells will activate certain factors within their cells at some point of development, which causes them to die. As you can imagine, scientists are very interested in this phenomena of apoptosis, as it seems to play a role in progressive neurodegenerative diseases, as well as a role in pruning out cells when we're still in utero. Diseases like Alzheimer's, Parkinson's disease, Huntington's disease, and Lou Gehrig's disease all have a portion of these diseases, the death of the important neurons that actually causes the symptoms of these diseases, apoptosis plays a role in that. It's not the complete role. It doesn't completely explain the degeneration of these neurons, but it certainly plays an important role. In these diseases, there's some type of initial insult to the brain that kills neurons and glial cells, but then later, sometimes over many years, many other cells in the central nervous system undergo apoptosis and contribute to the progression and worsening of the disease over time. So it's an interesting phenomena. It's also interesting because we would like to understand exactly how a cell basically commits suicide, because we would like cancer cells to do just that. So apoptosis is interesting for that reason as well. Is there some way that you could induce cancer cells to commit apoptosis? And obviously that would be very useful in the treatment of various types of cancers. The general consensus among scientists is that neurons and glial cells in the central nervous system no longer divide after the first few months of life. However, the process of synapse formation probably starts in the mid or late second trimester, and remodeling of synapses continues throughout the life of the individual. So, microscopic studies show that synapse formation is highest during the first 6 to 8 years of postnatal life. Then it plateaus and starts to decrease with the onset of puberty. This makes sense because the more synapses formed, the easier it is for the brain to acquire and store information. These first 6 to 8 years of life are when children have to acquire enormous amounts of information, including how to speak one or more languages, and also to learn countless rules, facts, and behaviors that make it a member of its culture. The brain's like a sponge for information during these years, and because the brain is continually forming large numbers of functional synapses at this time, it's no wonder that it's easy for a young child to learn several different languages at this time of their life, if they have the opportunity to hear these languages spoken on a regular basis. Peripheral neurons that have been crushed or severed are actually able to repair themselves to a significant extent, so that some or all of the function of the nerve can be recovered over time. A nerve that's crushed or severed in the central nervous system, however, is not able to repair itself, and the damage is typically permanent. Exactly why peripheral neurons can repair themselves, at least to a significant extent, if not completely, and central neurons can't, is the focus of much medical research. As you're no doubt aware, people who suffer spinal cord injuries and become paraplegics or quadriplegics won't recover from this injury with the science that we have right now. When these injured central nervous system neurons attempt to regenerate their axons, the process is halted and is never completed, unlike the case in peripheral neurons. It may be due to some inherent but as yet unknown difference between central and peripheral neurons, or it may be due to the release of factors by other nearby neurons or glial cells that inhibit CNS neuronal axons from ever actually being able to complete regeneration after spinal cord injuries. Spinal cord research, though, does offer the hope that someday scientists will solve this riddle, and a crushed or severed spinal cord will not automatically mean the rest of your life will be spent in a wheelchair. All right, when you're ready, go on to the next slide. Okay, we've now left section A of chapter 6 and are starting on section B, which essentially describes membrane potentials. This is an important chapter to spend some time on, as it explains the complex process whereby electrically excitable cells such as neurons are able to generate action potentials along their length and release neurotransmitters into a synaptic cleft. This figure, figure 6.7, shows that opposite charges attract each other and will move towards each other if not separated by some type of barrier. In addition, this figure shows that the electrical force of attraction between positive and negative charges increases with the number of charges present, as well as when the opposing charges get closer to one another. Electrolytes are chemical substances that separate when dissolved in fluids into electrically charged particles or ions. The fluid in our bodies, which can contain electrolytes or anything else for that matter, is either blood, interstitial fluid, or cerebrospinal fluid. The major cations, which are positively charged electrolytes found in our body's fluids, are sodium, potassium, calcium, and magnesium. The major anions, or negatively charged electrolytes in our body fluids, are chloride, phosphate, sulfate, and bicarbonate. You learned about ions in Chapter 4 when the issue of membrane potentials and separation of charges across cells was discussed. Here in Chapter 6, we'll spend more time looking at how this membrane potential is created and how neurons need to use membrane potentials to generate action potentials. For starters, it's a good idea to remember the concept of an electrochemical gradient, which you also first saw in Chapter 4. This concept's relatively simple. As we see here in this figure, electrical charges of the same sign repel each other, and electrical charges of the opposite sign attract each other. Thus, positive ions repel positive ions, and negative ions repel negative ions, but positive and negative ions are attracted to each other. Since the interior of most cells has a net negative charge with respect to the exterior of the cells, negatively charged ions want to leave cells and positively charged ions want to enter cells. On the other hand, you also learned in Chapter 4 that both ions and non-ionic compounds, like glucose, which is a non-ionic compound, want to diffuse across cell membranes towards the area of least concentration. Thus, the glucose molecules in solution with a glucose concentration of 100 millimoles per liter, separated by a plasma membrane from a solution with a glucose concentration of 50 millimoles per liter, will want to flow across the plasma membrane towards the 50 millimole per liter glucose concentration until the glucose concentration on both sides of the plasma membrane are equal. How much and how fast the glucose travels in the direction of the lower concentration is called the flux of the glucose. So, there are two things that make an ion or non-ionic compound want to enter a cell or not enter a cell: the concentration difference across the cell membrane and the electrical difference across the cell membrane, i.e. the membrane potential. These two forces together form what's called the electrochemical gradient across a cell membrane. Please take the time to read about Ohm's law here in this chapter and how the movement of electrical charge or current is a function of both voltage and resistance. Now let's go to the next slide and take a look at resting membrane potentials and how scientists can measure the membrane potential in a single cell. Okay, figure 6.8 shows how a voltmeter can be used to measure the resting membrane potential of a single neuron. Scientists can isolate a single electrically excitable cell, such as a neuron, under a powerful microscope and actually insert a tiny microscopic electrode into that cell, while the reference electrode is in a solution around the cell that mimics the extracellular fluid that's typically found around neurons. Once the microelectrode actually penetrates the neuronal cell membrane and enters the neuron, the potential difference across the cell membrane in millivolts can be measured. Remember, the potential difference is the difference in the amount of charge between the two electrodes. The asterisk in the bottom half of this figure shows the moment that the microelectrode actually penetrated inside the neuron. In this case, the potential difference measured was negative 70 millivolts. This neuron was resting, not firing, when this measurement was taken, so this is the resting membrane potential. When a scientist says the resting membrane potential in a neuron is negative 70 millivolts, what they mean is that it is 70 millivolts more negative inside the cell than whatever the polarity is of the extracellular fluid surrounding the cell. The extracellular fluid outside of the neuron is assigned a voltage of 0 just for simplicity's sake, and the membrane potential is simply stated as the sign of the excess charge that's inside the cell, either negative or positive. Electrically excitable cells have a resting membrane potential that's always more negative inside the cell with respect to net charge relative to the outside of the cell. So this particular study showed that the resting membrane potential was negative 70 millivolts, meaning that it's 70 millivolts more negative inside this neuron than in the solution surrounding the neuron. The reason for this is that the reason that the interior of cells is typically always more negative than the exterior is that the interior has many proteins and compounds with phosphate groups on them, which have a lot of negative charges attached to them at a pH of 7.4, which is the normal pH of biological fluids in humans. Most of these compounds were made inside the cell and are too large to be readily transported outside the cell into the extracellular space. So they remain trapped inside the neuron and give the cell interior a slightly more negative charge with respect to the outside of the cell. In neurons, a typical resting membrane potential ranges from about negative 40 to negative 90 millivolts. Now, go to the next slide and look at figure 6.9 and see how electrical charges across the cell membrane tend to collect together very closely near the cell membrane itself. Figure 6.9 shows how charged ions line up close to the cell membrane. This should make sense as these oppositely charged ions are attracted to each other. Therefore, the excess negative charges inside the cell are attracted close to the cell membrane, and the excess positive charges that are present on the outside of the cell also move close to the cell membrane. Remember, it's when these charges are allowed to travel across the cell membrane and come together that they generate an action potential and potentially cause a cell to become electrically excited. We'll see how this is accomplished in a short time here. A concept you need to keep in mind at this point is that the cell membrane itself is composed mostly of lipids, which do not allow these charged ions to readily cross back and forth from the cell interior to the cell exterior. The only way for these charged ions to travel across the cell membrane is through channels that span the lipid cell membranes. As you learned in Chapter 4, there are essentially three types of protein channels that form through cell membranes from the exterior to the interior: ligand-gated, voltage-gated, and mechanically gated channels. Opening and closing these channels for very brief moments allows ions to travel in and out of cells and change the resting membrane potential either up or down. As we will see shortly, when the interior of the cell becomes less negative due to positive ions flowing in, negative ions flowing out, or some combination of the two, we say the cell has become depolarized. When this happens, the cell may generate an action potential. If the cell interior becomes more negative as opposed to less negative, due to either more negative ions flowing in or positive ions flowing out, or some combination of the two, we say the cell is hyperpolarized as opposed to depolarized. When a cell is hyperpolarized, it is much less able to generate action potentials. So when a cell is depolarized, it's moving towards generating an action potential. And if a cell becomes hyperpolarized, it's moving away from being able to generate an action potential. Now let's go to the next slide.